Hello everyone, my name is Dr. Scott Howell. I'm the Research Director of Tier 1 Health and Wellness and a professor in the PhD Health Science program at Trident University. For those of you who don't know me or my background, my expertise is in androgen metabolism and advanced epidemiologic methods. So this video is part of a series in which I review key studies to demonstrate how literature should be interpreted by physicians and all those who consume research. If you go to PubMed, pull studies, if you do Google searches and pull studies that way, this is intended for you. If you consume research, watch these videos. Dr. Nichols is also reviewing studies and doing critiques as well. And I like to begin these presentations with interesting facts or statistics to get our minds thinking about statistical and research literacy. So in another video, I mentioned a piece by Wegworth, 2013, that noted that only 12% of 4,713 surveyed gynecology residents were able to correctly answer two simple questions on medical statistics, one on the meaning of a p-value and the other on the positive predictive value of a screening test. So think about this for a second. If most physicians do not understand the statistics in their own disciplines, then most should not be reviewing studies and making videos because this just adds to the overall confusion. My advice to physicians doing this is to gain statistical literacy and research literacy before reviewing videos as an expert on a particular matter. Not all studies have valid results, nor are they interpreted within the scope of the data. So you may ask yourself, why is this important to me? If most physicians do not understand the statistics, how confident are you in interpreting them? Ignorance is something that anyone cannot hide behind in today's information age, and most physicians, other professional doctorates, and laymen accept study findings at face value without question. But now more than ever, a critical but curious approach to all published research to gain statistical and research literacy is just good common sense. So over the past 5 years or so, I've seen a lot of false claims about androgens and how they work in the body. A lot of this pseudoscience stems from bodybuilding and some practitioners who do not specialize in this area. So the topic of this video addresses the myth of androgen receptor downregulation. And it's intended to get all of our followers up to speed on the real science about how androgen receptors are affected when someone's on hormone replacement therapy. So I'm very passionate about this topic because so many people have asked me whether increasing the dose of testosterone or an anabolic steroid leads to downregulation of androgen receptors. It's a never-ending issue that I've addressed for most of my adult life. And even several well-known TRT practitioners and some naturopaths have made this claim about downregulation within the last few months on YouTube. And one physician that used to be in a popular social media group used the term burnout to imply that higher testosterone doses desensitize androgen receptors by making them somehow less effective. So on the surface, this seems like a reasonable claim, but the problem here is that there is a fundamental ignorance of basic endocrinology and physiology when it comes to sex hormone metabolism and how these hormones are controlled through negative feedback. In the 1980s, this was the prevailing view in the scientific literature. And even Bill Phillips in the World Anabolic Review in 1996 made this claim, even though there was a compelling body of evidence that did not align and countered this view. So somewhere along the way, some clinicians and patients have equated downregulation of androgen receptors to the phenomena of tolerance that's seen with drugs of addiction. And I can assure you, all of our audience members, followers, that this is simply not true. Viewing this issue from a tolerance, saturation, absorption, or sensitivity perspective does not come anywhere close to characterizing what actually occurs in the body. So the questions that will be answered in this video include: what is the relationship between androgen receptors and negative feedback? What regulates androgen receptor expression? And by characterizing these two questions, we will completely answer the last question on whether increasing testosterone dose, or any androgen dose for that matter, downregulates androgen receptors. So everyone should know the history of testosterone. If they're taking hormone therapy or if they're giving advice to someone who is. The majority of you probably know that testosterone was synthesized in 1935, but most of you probably don't know that one of the first two synthesis processes was a methylation synthesis to derive methyl testosterone from cholesterol. And this occurred when Ruzicka attempted to mimic the natural synthesis pathways in the body. And although Ruzicka published his synthesis a week after Butanant, his synthesis of methyl testosterone allowed oral absorption to bypass second pass metabolism without intending to do so in the beginning of the history of synthetic androgens. So this is why testosterone is not an anabolic steroid. It was after the initial 17-alpha alkylated synthesis that researchers began to try to dissociate the androgenic effects from the anabolic effects. And many scientific papers of authors that should know better have applied and reported a post hoc conception of anabolic steroids to include testosterone, but this is simply not correct. Testosterone was and continues to be the yardstick by which anabolic steroids and SARMs are measured. So the errors in historical content go back even further. Ruzicka and Butanant did not specifically receive Nobel Prizes for the synthesis of testosterone, like what has been published in many papers or on many websites. Butanant received the Nobel for his work in identifying the molecular structure of sex hormones, and Ruzicka received the Nobel for his use of polymethylines and higher terpenes, and not only the synthesis of sex hormones, but also to synthesize perfumes. So why does this matter to androgen receptor downregulation? Well, from 1935 to the present day, 84 years have elapsed, and androgens became one of the most extensively studied drug classes in existence. So the answers to many of the questions, such as those we are answering now in this video, have been known for quite a long time, but apparently no one digs deep enough to know the literature. So now we turn our attention to negative feedback loops. The issue of equating the control of androgens to the concept of tolerance is a fundamental ignorance of the basic homeostatic model on this slide and how tolerance really works. For many physiologic processes like the regulation of body temperature, this model fits very well. A receptor senses a change, the receptor signals to a control center, and then the control center initiates action in an effector to increase or decrease a physiologic parameter. So essentially, we have three primary components in negative feedback: a receptor, a control center, and an effector. And each of these components works to keep a hormone or physiologic process in homeostasis within a certain range conducive to maintain life. Now we look at the HPG axis. The hypothalamus pituitary gonadal axis has 4 points in which negative feedback occurs, similar to the homeostatic model that we looked at on the last slide. The kisspeptin system acts like a sensor. The hypothalamus acts as a control center, and the gonadotrophins function as effectors, and the Leydig cells function as the effector cells. The set point for testosterone is critically important because set points for physiologic processes can change given certain circumstances, such as an infection. With the infection analogy, the body compensates by increasing the set point for temperature. And this is why you run a fever, and when the fever breaks, the set point resets to a lower value. Now, depending on the set point, peripheral levels of testosterone, other sex steroids such as estrogen and progesterone, along with metabolic signals like leptin, are sensed by the kisspeptin system in the arcuate nucleus of the hypothalamus, in the control center. And this stimulates KISS1 expression. Now, this in turn stimulates GnRH neurons to secrete gonadotropin-releasing hormone into the portal pituitary circulation. And then GnRH binds to its receptors in the pituitary, stimulating the secretion of the gonadotropins into systemic circulation. The gonadotropins include the effectors known as luteinizing hormone primarily and a follicle stimulating hormone, FSH, to a lesser extent. So luteinizing hormone, or LH, then stimulates the Leydig cells in the gonads to produce testosterone. So ask yourself a question. Where are androgen receptors controlled in this negative feedback? Although androgens stimulate the kisspeptin system, other sex steroids and metabolic hormones also stimulate the system. Androgen receptor binding acts only through the AR-DNA binding domain to repress gonadotropin releasing hormone transcription as one of the many regulators of GnRH release. The entire process has nothing to do with androgen receptor expression in target tissues. Androgen receptors are not controlled by negative feedback, and for good reason. Before we jump into how androgen receptors are regulated, it's good for us to visualize what we're talking about and see how complex the androgen receptor and its actions really are. The androgen receptor is a protein made of 919 amino acids in healthy humans, but those with polymorphisms, the number may go as low as to 910. The androgen receptor contains a ligand binding domain, and this is AF2 at the top right in green, a DNA binding domain at the top center in light orange, and an amino terminal domain, AF1 at the top left in cream color. So obviously the natural hormone agonists of the androgen receptor are testosterone and the 5-alpha reduced metabolite dihydrotestosterone. So now we look at the classical model of androgen receptor action. This is the model taught in medical school and presented in most physiology texts. In the absence of androgens, the androgen receptor is located in the cytosol of the cell as part of a multi-protein complex with chaperones. They include heat shock protein 90 and heat shock protein 70. And the co-chaperone, FKB P51. When either testosterone or DHT diffuses through the plasma membrane and binds, this causes conformational changes in the androgen receptor protein complex. Binding permits the dimerization or adducting of the androgen receptor in what is referred to as an NC-terminal interaction. Then the complex translocates to the nucleus where the receptor dimers are stabilized, and binding to the DNA disrupts the NC-terminal interaction. So this opens up the region of the androgen receptor terminal domain for interactions with cellular transcriptional machinery. The machinery includes many co-regulators that recruit RNA polymerase to create messenger RNA that eventually gets shuttled back to the cytosol to undergo translation at the ribosomes to produce functional proteins. The important point here is that the process from binding to functional proteins takes hours to demonstrate a physiologic response. Non-classical androgen receptor action works considerably faster than the classical model. To understand this, graphic, it's helpful to know that because the androgen receptor functions as a transcription factor, our molecular understanding of the role of testosterone has been established through the identification of androgen receptor target genes. These genes were identified due to the presence of androgen response elements, or AREs, within the promoter regions. Due to all the steps, changes in gene transcription can take several hours to show a physiologic response in the classical model. However, in 2004, isolated Sertoli cells were shown to rapidly respond to testosterone stimulation in less than 1 minute by upregulating the activity of MAP kinase signaling, which was found to be dependent upon the androgen receptor. The seminal work by Tuchek 2016 used specific activators and inhibitors in both classical and non-classical signaling to show that both pathways shared overlapping functions that retained distinct roles in spermatogenesis in the Sertoli cells. In non-classical AR signaling, testosterone stimulation transiently increases androgen receptor localization to the plasma membrane. And this just means that they start to aggregate towards the membrane. They localize, they move towards it. And this results in localized androgen receptor activating SRC tyrosine kinase. SRC activation in turn alters phosphorylation and activation of EGF receptors that additionally activates the MAP kinase cascade, RAS, RAF, and ERK. Further signaling through P90 kinase causes phosphorylation of the Krebs transcription factor, resulting in an increased transcription of Krebs-inducible genes. Now, armed with our knowledge of both slow and fast androgen receptor actions, we look at evidence of how androgen receptors are regulated. I will use the term upregulation, but in reality, androgen receptors are auto-regulated or self-regulated at the tissue level of control. And this has very important clinical implications when we talk about dose response. So even before the research that you see on this slide, there was a substantial amount of literature that basically hypothesized that there would be downregulation, but in the published write-ups, there was actually evidence of upregulation. Many other researchers believed at this time that downregulation would occur and attempted to point to various mechanisms for the contradictory results that did not meet their hypotheses. So Gadd in 1998 observed upregulation of androgen receptors on genital skin fibroblast upon prolonged exposure to DHT and an anabolic steroid, metribolone. Metribolone is an oral form of trenbolone that is 17-alpha alkylated and was referred to as research chemical 1820 or 1881. At 20 hours of exposure to both hormones, there was a 2.07-fold increase in upregulation by DHT and a 2.35-fold increase with metribolone. The authors concluded that upregulation plays a role in the high affinity binding of androgen receptors in the nuclear matrix. Gonzalez in 1993 looked at smooth muscle cells cultured from immature and adult rat penises to assess downregulators of AR expression as a function of cell proliferation in smooth muscle of androgen responsive vascular tissue. They applied metribolone to each cell type, and they found that contrary to their hypothesis that androgen receptors would be downregulated, the androgen receptors were upregulated in adult penile tissue. When they prevented the contact of the hormones in the medium they had and depleted the serum of androgens, this downregulated AR expression levels, but upon re-exposure with DHT, the downregulation was countered. So here we have the beginning of a dose response. Dumet in 1996 investigated whether androgen receptors existed in skeletal muscle satellite cells and myotubes and they looked at the effect of testosterone on satellite cell proliferation and differentiation. Here, proliferation just means to increase the number of a single cell type, and differentiation means that the cells have to take a lineage. They have to be directed into a type. Here, these cells could differentiate into either myocytes or adipocytes. The researchers found that in both satellite cells and myotubes, AR increased at each measurement increment at 6, 12, and 24 hours of exposure to testosterone. The authors concluded that satellite cells were direct targets of androgen action, and testosterone administration increased the number of androgen receptors. So this study by Wyron and researchers looked at exposure to DHT and AR expression in human osteoblasts or bone cell lines. DHT increased androgen receptor expression in a time and dose-dependent manner. And interestingly, when an anti-androgen was applied, it attenuated the upregulation. So the authors concluded that physiological concentrations of androgens had substantive effects on androgen receptor expression in bone tissue. So we also have a upregulation in bone tissue as well, another target tissue. So this study by Lynch assessed upregulation of androgen receptors with several anabolic steroids and DHT. They injected 2 milligrams per kilogram of testosterone cypionate or nandrolone decanoate, 1 milligram per kilogram of boldenone or 5 milligrams per kilogram of dihydrotestosterone into castrated rats. After 18 to 24 hours, they sacrificed the rats and excised the brain tissue, and they found that each of these androgens upregulated AR in the ventromedial hypothalamus, the medial amygdala, and medial preoptic areas. So now we have penile vascular tissue, we have bone tissue, and we have brain tissue where AR has been upregulated. So this study by Hankey looked at the mechanisms behind protective cardiovascular effects of testosterone in rabbits, and they found that AR upregulation also occurred in major vascular tissues. And this is very, very important. Not only did testosterone inhibit new intimal plaque development, so the intima is a layer of vessels. So testosterone inhibited plaque development in this layer, but it also increased AR expression by 50% in each of the arterial segments that were assessed. So despite the claims that nitric oxide release is the primary cardiovascular benefit of therapy, and testosterone therapy, I argue that direct AR action is the primary benefit of optimization therapy in androgen responsive tissues. Nitric oxide is just one of the many benefits of direct androgen action in the vasculature. So specifically AR action to prevent new intimal plaque development, to reduce adhesion, molecule expression, to decrease inflammatory cytokines, to favorably modulate the lipid profile, and to increase vascular reactivity, and to mediate nitric oxide release. And the vascular reactivity is probably one of the most important cardiovascular factors of testosterone therapy, the biggest benefit. These are the primary cardioprotective benefits of testosterone, and there's not many people that talk about these. So this study also looked at vascular tissue, specifically aortic vascular smooth muscle cells of rats. At first, the researchers characterized the time response from 0 to 48 hours of testosterone exposure, and they found androgen treatment for just 10 minutes induced a transient downregulation of cytoplasmic androgen receptor expression. But afterwards, there was a gradual increase in androgen receptor expression that plateaued at a 5-fold increase at 48 hours from baseline. Nuclear androgen receptor increases became evident at 24 hours, but reached greater AR expression at 48 hours. The researchers concluded that androgen exposure to physiologic concentrations increased AR expression in vascular smooth muscle cells at the level of transcription and non-transcription. This reconfirms the mechanisms for protective cardiovascular effects we mentioned on the previous slide, that direct AR action coupled with fast signaling increases vascular reactivity, prevents new animal plaque development, lowers inflammation, modulates the lipid profile, and stimulates the release of nitric oxide. So vascular reactivity, remember that. This study by Carson and researchers looked at AR expression with high-dose nandrolone, decanoate, and rat hind limbs. They used 6 milligrams per kilogram, which equates to roughly 545 milligrams per week in a 200-pound man. They found in the soleus muscle, nandrolone increased AR expression by 250% in 5-month-old rats and 900% in 25-month-old rats compared to control rats of the same age. So here, nandrolone increased AR expression with increasing age, likely as a compensatory mechanism. So we see an interesting finding of an effect modification by nandrolone in age to increase androgen receptor expression. So this study by the Porcine group demonstrated that androgen receptors were upregulated with both DHT and testosterone and mesenchymal pluripotent stem cells of mice. Now, these somatic stem cells have 2 options for differentiation, the same that we mentioned before. They can take either a myogenic lineage into muscle cells or an adipogenic lineage into fat cells. So here, the researchers not only provided more evidence of upregulation, but also showed that DHT and testosterone regulate stem cell lineage determination by promoting mesenchymal pluripotent stem cells to differentiate into muscle cells instead of adipose cells. Now, this is another unmentioned positive effect of testosterone therapy that no one talks about, and it explains in part how body recomposition occurs in individuals under androgen therapy. And there's some new evidence to suggest that micro RNAs are very important to this process. So now we move from animal studies to human studies. And here, Cotty and researchers assessed 8 high-level powerlifters without anabolic steroid use, 9 high-level powerlifters with anabolic steroid use, and 6 untrained subjects as controls. The intent was to determine whether long-term strength training and self-administration of anabolic steroids independently and together acted to change androgen receptor content in certain skeletal muscles. So after biopsies in the trapezius muscle, they found AR-containing myonuclei were higher in non-using powerlifters compared to controls. But they also found that anabolic steroid users had higher AR than both non-using powerlifters and controls. In the vastus lateralis, the myonuclear number was also higher in the anabolic steroid using powerlifters compared to non-using powerlifters and controls. So the researchers concluded that AR increases differed by muscle type according to strength training, which is an independent stimulus, in anabolic steroid use. And these effects were completely independent of one another. This is another line of evidence showing autoregulation given different muscle types at the tissue level of control. So now, tying all this together up to this point, this study by the Porcine group in 2004 assessed biopsy samples of 6 healthy men that were administered 600 milligrams per week of testosterone enanthate over a 20-week period. Most of us that are in this field know these as seminal studies, and many But there are some people that believe that there were multiple trials, but in actuality, there's only 2 or 3 trials that were actually conducted with this high of a dose. So building on this, on the study presented in the previous slide, the researchers sought to determine which muscle tissue cells expressed upregulated androgen receptors, and they found that AR were expressed predominantly in muscle satellite cells, myonuclei and myofibers, CD4 stem cells in the interstitium, fibroblast expression in the endothelial and smooth muscle cells, and mast cells also expressed higher androgen receptor content. And they also found higher androgen receptor expression in endothelial and smooth muscle cells, and concluded that high dose of testosterone increased androgen receptor expression in satellite cells and myonuclei while increasing nucleolar AR density. So now we see that not only physiologic doses increase AR expression, but we have a dose response. We have a graded response, and even up to super physiologic doses, which I hate that term, androgen receptors keep increasing. So I included this slide to demonstrate how science proceeds in the academic literature and how researchers must change their beliefs in the face of evidence. So we just saw the Porcine's research group that found greater AR expression in one of the supraphysiologic studies that used 600 milligrams per week in healthy men in 2004. And now you see what the very same research group claimed in 2001. Here, 3 years before, they had found higher AR expression in many muscle cell types. They stated verbatim that androgen receptors in most tissues are either saturated or downregulated at physiologic concentrations. They cited 3 animal studies and 1 review as evidence for this claim, and I was very disappointed to see a primary research study that referenced a literature review as evidence to support this claim. So we have established in this short video that the evidence during that time did not warrant this claim. And it should be clear by now to all of you that any instance of saturation would only be transient, because autoregulation of the AR gene shifts continuously in a dose-responsive manner at the tissue level of control. And this means that for saturation to occur, it would have to do so in many target tissues simultaneously, which we know does not happen based on the evidence we have seen so far. This is why the word saturation used to describe AR binding is almost never used in the correct context and has no real meaning to characterize the general process of AR binding or expression. So I decided to include this slide to show the clinical importance of autoregulation in other fields of medicine outside of testosterone replacement. So specifically, how errors in autoregulation result in a paradoxical effects in AR expression, specifically in castration-resistant prostate cancer. So here, instead of downregulating AR expression in response to castration levels of androgens, as would be the case in normal conditions in healthy patients, castration-resistant prostate cancer cells adaptively autoregulate AR expression by increasing androgen receptor content to a 25-fold or greater number in low androgen environments. Upon exposure with metribolone in this study, prostate cancer cells did not proliferate as seen with controls, or to a lesser extent with anti-androgen exposure. Here, exposure to metribolone lowered prostate cancer cell number as shown in Figure A, or attenuated cancer cell proliferation as shown in Figure D. Now, this paradox shows why bipolar androgen therapy is used to treat castration-resistant prostate cancer. Normally, androgen receptors would increase with increasing androgen dose, but here the opposite occurs. When androgen levels drop, androgen receptor expression increases. So to answer the first question that we posed in the beginning of this video, what is the relationship between androgen receptors and negative feedback? Well, although endogenous androgen levels are controlled by negative feedback, androgen receptors are not controlled by this mechanism. AR expression is autoregulated at the tissue level of control by way of the AR gene. The AR DNA binding domain acts as a repressor of gonadotropin-releasing hormone transcription as one of many other hormones that regulate the release of GnRH in the HPG axis. The HPG axis has nothing to do with AR expression in target tissues. So what regulates androgen receptors? Well, so far, we have seen that physiologic concentrations and high dose concentrations of testosterone, DHT, metribolone, and other anabolic steroids upregulate androgen receptors in penile tissue, penile vascular tissue, arterial tissue, and vascular smooth muscle cells, brain tissue, satellite cells and myofibers and muscle tissue, and several osteogenic cells in bone tissue. We see that the effect is dose responsive, meaning that as androgen levels decrease, the androgen receptor gene autoregulates by decreasing androgen receptor number, and vice versa. When androgen levels increase, the androgen receptor gene autoregulates by increasing androgen receptor expression. We have seen how errors in autoregulation are involved with castration-resistant prostate cancer, and we see that as androgen dose increases, the androgen receptor gene autoregulates by increasing AR expression. Now, this is the primary reason that anabolic steroids used with extremely high doses continue to work well and be effective, because the body compensates for the increased androgen load by stimulating the AR gene to produce greater numbers of androgen receptors in the target tissues. Despite what some of these individuals are saying in other channels about that there's a threshold regarding dose and that the androgen receptors are desensitized, this simply does not occur. There's other reasons that people plateau in this situation. Androgen receptor expression is controlled by the process of autoregulation independent of negative feedback. All sex steroid receptors have some evidence of control by autoregulation, although the literature is not clearly demarcated on this process outside of the androgen receptor. So contrary to recent claims, increasing androgen dose to any level does not burn out or downregulate androgen receptors. So finally, we definitively answer whether androgen receptors are downregulated with increasing testosterone dose, and the answer is a resounding no. And this has been known at least for the past 30 years. So I want to thank each of you for watching this video and doing your part to become an educated and literate consumer of research. Be sure to hit the like button and follow us on YouTube, Facebook, Twitter, and Instagram. Come and visit us and see evidence-based practice in real time. Reach out to us with any topics that you would like to hear about or other questions related to hormone replacement therapy or anything else that involves men and women's health. Thank you.